Hello students, today our topic is recombinant DNA technology. Let's study this topic in detail. So let's begin our class regarding recombinant DNA technology. The technology used for producing artificial DNA through the combination of different genetic materials from different sources is referred to as recombinant DNA technology. Recombinant DNA technology is also popularly known as genetic engineering or gene splicing. It is defined as a set of molecular techniques that allows the manipulation of the DNA to create new combinations of genes, often obtained from different microorganisms. This technology has a profound impact on various fields like medicine, agriculture, industry, and etc. The recombinant DNA technology emerged with the discovery of restriction enzymes by the Swiss microbiologist Werner Arber. Inserting the desired gene into the genome of a host is not as easy as it sounds. It involves selection of desired gene for administration into the host, followed by a selection of the perfect vector with which the gene has to be integrated, and recombinant DNA is then formed. Thus, choosing a perfect vector and host plays a vital role in this technology. Thus, the recombinant DNA technology has to be introduced into the host. It has to be maintained and carry forward to the next offspring without getting altered. So, what are the various tools or efficient enzymes that are used in recombinant DNA technology? These are the following enzymes which plays a vital role in this technology. The first one is the restriction enzymes. These enzymes are the endonucleases which cuts the DNA at specific sites. The second are the DNA ligases. Ligase joins the DNA fragments. DNA polymerases are used in the synthesis of the DNA strands. They are even used in PCR techniques as well. Transcriptase enzyme. These enzymes converts the RNA template to DNA template. They are used in cDNA synthesis as well. Alkaline phosphatase enzymes. These enzymes remove phosphate groups from the DNA. Topoisomerase enzymes. These enzymes removes the tension created in the DNA by causing temporary breaks and rejoining, thus manipulating the DNA topology. Nucleases enzymes. These enzymes are used to remove unwanted DNA or RNA sequences. Thus, all these enzymes facilitate work together in gene cloning, PCR techniques, and other processes in the genetic engineering. Before starting the recombinant DNA technology process, a host organism is considered. A host organism is the one in which the recombinant DNA is introduced. The host is the ultimate tool of DNA technology, which takes in the vectored engineering with the desired DNA with the helps of the enzymes. There are a number of ways and steps in which these recombinant DNAs are inserted into the host. Namely, it could be microinjection, biologist, or gene gun, or alternate cooling and heating and use of calcium ions, etc. So now let's look in detail the various steps involved in process of recombinant DNA technology. The first step here is the isolation of genetic material. The first and initial step in recombinant DNA technology is to isolate the desired DNA in its pure form. That is, it has to be pure and free from all macromolecules. Okay. Once the DNA is extracted, then cutting, the second step is the cutting the genes at the recognition sites with the use of restriction enzymes. The restriction enzymes play a very important role in determining the location at which the desired gene is inserted into vector genome. These reactions are called as restriction enzyme digestion. So, the here the desired gene is introduced into the vector genome. The third step is the amplification, the gene copy through polymer chain reaction. Once the desired vector genome is obtained, it is amplified through the PCR techniques, and then addition of the ligase to seal the DNA with covalent bonds. Okay, this is called ligation of DNA molecules. And the fifth step is that this plasmid is injected into a bacterium by a technique called transformation, that is insertion of recombinant DNA into host. In this step, the recombinant DNA is introduced into the recipient host cell. This process is called as transformation. Once the recombinant DNA is inserted into the host cell, it gets multiplied and is expressed in the form of the manufactured protein under optimum conditions. As mentioned, there are various ways in which this could be achieved. The effectively transformed cell or organisms carry forward the recombinant gene without getting altered uh, or exercised to its offspring. Okay? Thus, the process of recombinant DNA technology and its simplified steps. So, what are the applications of this technology? There are various applications like bacteria, yeast, plants, all could be genetically modified to produce important pharmaceutical enriched foods and industrial important products. Recombinant DNA technology has its application in diagnostic purposes, like it is used to detect the presence of HIV in a person. In the field of medicines, recombinant DNA technology is used for the production of insulin. In clinical diagnosis, ELISA is one of the best example which uses the technology of recombinant DNA technology. Recombinant DNA technology is widely used in agriculture to produce genetically modified organisms such as flavor savar tomatoes, um, golden rice rich in proteins, BT cotton to protect the plants against various phytopathogens, etc. And gene therapy, it is used as an attempt to correct the genetic defects which gives rise to hereditary diseases. Okay, in environment, it has its application in the form of biosensors, genome editing, and recombinant DNA cloning can be some of the possible applications of the recombinant DNA technology. So students, we are done with the basic introduction of recombinant DNA technology. Hope this class was beneficial to you. Thank you.